So, hello and welcome to another episode of Interviews with Experts. I'm Frederick Dunn, and this is The Way to Be. Varroa mites are moving freely through our beehives and reproducing in brood cells while they pass on a range of pathogens that are extremely detrimental to our honeybees. Spider venom just may be the next best tool against this parasite. Today, my very special guest is Dr. Volker Herzig. Dr. Herzig is an Australian-based researcher and arachnid expert at the University of the Sunshine Coast. He's known affectionately in scientific circles as Spider-Man. He houses the world's largest collection of arachnid venoms, comprising more than 870 spider and scorpion venoms, many of which he was the first to analyze. Here's Associate Professor Volker Herzig. So, hello and welcome. I want to thank you for joining me on The Way to Be. And Dr. Herzig, I really appreciate that you're taking the time to talk to us all the way from the other side of the world. And if you would, please, we do have an audience that's pretty spread out. We're primarily backyard beekeepers and researchers and backyard citizen scientists. And so one of my viewers actually asked me to reach out to you, and I really appreciate that you responded and that now you're here to share what's going on in your part of the world. So if you would just take a minute to introduce yourself to the listeners and describe where you are and what you're doing right now for work.
Okay, yeah, it's my pleasure to talk to you and to your listeners. So yeah, I'm talking from Australia, from the Lockyer Valley at the moment. This is where I live. And just to give you a little bit of background on myself, so I was born in Germany, and then I studied like biology in Germany and did my degrees there. So I got a PhD in biology from a German university in Tübingen. And then that was in 2004. And then I did a postdoc in Australia. So that was in 2005. I started my first postdoc. So postdoctoral research, which is what you normally do after finishing your PhD. So that was at Monash University in Melbourne, where I was for 3 years. And after that, I have joined another sort of famous researcher in this field, which is Professor Glenn King. So he's basically the leading authority on spider and other arthropod venoms in the world. And so he started his lab in Brisbane back in 2008. And then I joined his group then, and I stayed in his lab as a postdoc for about 12 years. And then in 2020, I got a future fellowship here from the Australian Research Council, and that allowed me to start my own research group at the University of the Sunshine Coast. And this is where I've been since. But all the time since I'm in Australia, I'm basically, doing research on spider venoms. And then after a few years, I also sort of branched out a little bit into scorpion venoms as well, and some other, like insect venoms, assassin bugs, and excuse me, and centipedes. But it's mainly arachnids, so mainly spiders and scorpions that interest me. And I've always been interested in spiders since I was a little boy. So I've, at the house of my parents in Germany, and I was feeding the orb weaving spiders that are around the house with locusts, and watching them how they wrap up the spiders, so they wrap up the locusts and eat them, and so that I was always fascinated by that. And yeah, I guess even from early childhood, like my yeah, my further way was already determined, kind of, to become a researcher in doing something with spiders, and eventually, it worked out to be in the field of toxinology. So I was interested in their venoms and toxins. Yeah, that's sort of the background from myself.
Okay, so I have to ask right off the bat, since this is a childhood thing as well, that you were deep into arachnids and you watch orb weavers kill their food, eat their food, and of course how they catch it. Did you keep any as pets inside your house, and do you keep any now?
So no, I didn't keep orb weavers, but so in Europe it's pretty popular keeping tarantulas, and over the last few decades it has become even more popular. But Yeah, I did keep some tarantulas as pets, and it was a bit funny. So that was when I was, like a teen. I don't know, maybe around 13, 14, when I wanted my first tarantula. But it took me about 2 years to convince my parents to allow me keeping the tarantulas because they had their reservations. They were like scared of tarantulas, spider venom, dangerous, whatever. But eventually, I managed to convince them, to have one, and I got it as a little baby, and then I could watch it grow. Um, and then eventually, it's a bit like collecting stamps. It never stays with just one. So then you need to get another one and another one. And there's so many different species, and all of them, or many of them, are beautifully colored. And you want to have them all. So eventually, I had a few dozen tarantulas
A few
in my
dozen.
room.
Okay.
And yeah, and when they make babies, that can be like 500 to 1,000 babies. So, I mean, not counting them, just the larger ones were
Yeah.
a few dozen. And then there's trade fairs, every, year or so, or every half a year, there's trade fairs in different places. So you can go there and trade your spiders for some others. And it's a pretty interesting scene. And yeah, that's where I got into as a teen. And what was really funny about it was that um, so initially, I said my parents had the reservations, me bringing in the spiders. So eventually they agreed, and then every visitor they had, who was visiting them, they brought to my room, and then, oh, can you show them the spiders? And then I started explaining and showing them, and they were always standing there listening to my explanations. And then after a few years, my parents were actually doing the explanations. So I was just sitting there, and they were bringing the visitors, and they were explaining, oh, this is this spider from here, and this whatever can do this. And so it's, yeah, I mean, they basically learned a lot about the spiders and forgot their initial fear. And yeah, I just found it funny, that they were eventually doing the guided tours through my spider collection, and I just was sitting there enjoying myself.
Yeah, look, what a debt you owe to your parents for ultimately allowing you to keep them. That way you didn't have to hide them somewhere in the closet or under your desk, as long as you had
Yeah,
permission.
that was great. I very much appreciate that. Yeah.
Yeah.
And as to get back to your question, so now I'm not keeping any tarantulas or any other spiders as pets. I mean, we do have a few larger orb weavers, in our backyard here, especially around the chook pen, because chooks poop a lot, and that attracts the flies, and that obviously attracts a lot of orb weavers. But
Okay,
yeah, I don't keep them.
I have to clear that up for the American listeners. A chook is a chicken.
Oh
So
yeah, yeah, sorry.
we're talking about, they call them chooks there, but we call them chickens. All right.
Chicken,
So
yes.
yeah. So you see the orb weavers around your chicken coops and pen areas and stuff like that.
Yes, yes. So we have those, we call them golden orb weavers. So that's a
Hmm.
spider that has maybe, 3 or 4 centimeters body length. And with the
Wow.
legs,
Yeah.
some of the bigger ones with the legs, they can always be the size of your hand, but they're pretty harmless. So they only have toxins in their venom that can harm insects.
Yeah.
But for humans, they're basically harmless. They look a bit scary, and it's more awkward if you walk into one of their webs, because like their orb webs, they can be, like a meter or more in diameter, and they're pretty
Oh,
solid.
yeah. Yeah.
So when you walk in there, you kind of get stuck
Yeah.
almost. And I have seen sort of photos and videos where like a bird or a bat has flown into these webs
Mhm.
and it got stuck and then wrapped up by the spider and eaten. So these and there are some anecdotes that in some island folks, they have used these webs actually for fishing, because that's like they're so sturdy, and they just
Yeah.
use a stick and collect, like bend it to a circle, and then collect maybe 10 or 20 webs, and that's sort of like a little bit of a fishing net, and then they catch small fish
That's
instead.
remarkable. Wow.
Yeah.
Well, that's already more than a bargain for. We have a lot going on here.
It's
Um,
Yeah, that's a bit distracting, obviously, from the Venom story.
Yeah, no, but it's very interesting to me personally, and I just hope that that falls on others too, that they just want to learn in general. A lot of people don't understand how long a tarantula can live. Like, what is the longest lifespan of a large spider like that?
So tarantulas can live up to 20 or sometimes 25 years. But this is, data from captivity. Because obviously, in captivity, I mean, you do care a lot of your spider. If there's some issues, you try to protect them from predators and whatever. So I think in nature, they might only be maybe around 10 years old, maybe 15 if they're lucky. But once they get older and slower, they might not be able to catch prey anymore as efficiently. So and then or they might be more easily captured by predators. Whereas in captivity, you can control that. Obviously, you don't have any predators, and you only give them food, when they're really hungry. Because, for example, if you feed them like house crickets, so they also can start nibbling on the spider if the spider doesn't eat them. And they eat each other, the crickets, but they can also eat the spider, especially if the spider molts. So if it changes, sort of gets out of the old exoskeleton to grow, um, that's a phase where they're pretty, how do you say,
They're
um,
vulnerable.
susceptible, yeah, susceptible to predators. And because the exoskeleton, the new one, is quite soft, and if you still have like feeding insects inside your terrarium where you keep them, it could potentially eat a spider. But normally you control that as a pet keeper. But in the wild, that can't be controlled that well. So I think in the wild, maybe 10, 15 years might be the max. But yeah, in terrariums it's over 25 years. Although there is actually a report from a trapdoor spider in Australia, and they monitored it in the wild for, I think it was over 40 years. So there are even spiders that get older than the tarantulas. So I think that's the oldest recorded spider is one
So
from
I
Australia.
so I have to ask, some spiders are, migrating around the world. We have the Joro spider here that makes a huge web that you're describing. I don't know if you know anything about that, but it's
So that's
expanding.
actually the same as the golden orb. So they belong
Oh,
like to the same genus. So we call them golden orb because if you look at the web actually has a little bit of a yellow color, or
okay.
golden, you could say. And that's why they call it the golden orb, because the web is a bit yellowish colored. But a choro spider is the same, it's Nephila. Yeah, but I mean
Okay.
they are harmless. They're totally harmless to
Right.
humans.
They're but visually impressive, and then that big web
Yes,
that you described, is that the webbing that you're saying people were using for fishing line? Is it that?
yes.
Yeah,
That's the
okay.
one. Yeah.
Now it's all making sense. All right, I'm catching up. Okay, so, and I have to ask because I know somebody's probably looking at the picture behind your head there. Is that that cave spider? What is that?
Yes, that's the Tasmanian cave spider. I can go down a
Tasmanian
little
cave
bit.
spider. If ever a spider had a friendly name, Tasmanian cave spider. So, does it
Yeah.
live in the dark? Would it have small eyes? What's going on with that specimen?
So yeah, it actually lives in only in caves, so it needs that sort of constant, like temperature in the caves. And I mean, in general, like the eyesight of most spiders is not very good, except for example, jumping spiders. I mean, you will notice, when you see a jumping spider, I mean, it has this huge like pair of front eyes, and if you move, it will sort of turn around and look at you.
Sure.
But normally other spiders, for them, like the eyesight is not that crucial. It's more like other senses, like vibration, for example, is a very important sense, especially for orb weavers, because they sit in their web and they
Mhm.
can sense any tiny vibration in the web, and then that guides them to where the vibration comes from and where the prey is. And so the sense of vibration is a lot more important to most spiders than the eyesight. But there are some exceptions of some spiders that hunt in daylight, um, like the jumping spiders. They need their eyes, but normally like eyesight is not that crucial for spiders, and especially not for the cave spiders, obviously.
Right. Okay, so I have to ask this because obviously the end discussion here is to talk about how you're using venom properties to control the Varroa destructor mite in honeybees. But did you happen to start with spiders that you already kind of have as favorites to evaluate their venom for that purpose? Or how many species do you have to go through to figure out which one is going to produce the venom that has the traits that you need for the Varroa destructor mite?
Um, so the way that works, and that this is not just for for our screening against the varroa mite, because in my lab, that's not the only target species we're looking at. So
Mhm.
we are looking basically at any kind of pest insect or pest invertebrate, that is relevant for agriculture. We have like we screen against some flies which are pests or beetles, also caterpillars, so the
Mhm.
larvae of butterflies and moths, um, and locusts. More recently, we have a program on screening toxin venoms against locusts to identify the active toxins. And for all of these programs, it never works like I have a favorite spider and I'm looking at that venom. So it's more kind of a blinded study. So we pick a number of venoms. Let's say we start with 100 or 200 different venoms, and I try to make that panel for the testing as diverse as possible. So I don't just look for tarantula venoms. I look for other venoms that we might have in the collection as well. So like huntsman spiders or funnel web spiders, and then I also include some scorpion venoms as well from different families, to have a really complex selection of these venoms. And then we test them all one after the other in our target species, so whether it's the varroa mite or the locust or whatever we are screening. And then we look at the results. So which venoms performed the best? And normally we get like a hit rate of maybe 10 to 20%. So if we screen like 100 venoms, we might end up with 10 or 20 that worked quite well. And then we look more closely. So what are the 10 best venoms? So like if there's some that are from related species, we only pick one rather than doing, because it's very likely if you have venoms from like three related species, they will all have very similar compounds. And we're looking to maximize the diversity of compounds that we can isolate. So we rather look for diverse selection of hit venoms. And yeah, then we pick the best ones based on those considerations, but also based on the venom availability. So sometimes, if we don't have enough of a venom to follow up, there's no point, in selecting that one. So we pick the one, if we have the choice, there's a venom from the same genus and like two different venoms from the same genus, and of one we have like 1 milligram, and of the other one we have 10 milligram, then we pick the one where we have 10 milligram, because that more likely gives us enough material to follow through all the way until we have isolated and identified our toxin. And so this is sort of the initial process of doing the venom screens. So you have a target species like the varroa mite, and you test a whole bunch of venoms, and then you pick the ones that work best. But it doesn't end there. So that's only the start of the challenge, because these venoms, they are really complex chemical mixtures. So they are not just, like two or three compounds in there. So some of the venoms that we analyzed, have more than 3,000 different compounds in them. And only one of them might be the one that you're looking for that's active against the varroa mite or doing whatever you want it to do, which means you need to select that one and isolate it from the other 3,000. And that's actually the challenge. And we have like what's called an high pressure liquid chromatography, or high performance liquid chromatography, HPLC. And this is a way, where we can fractionate the venom into different components. And this is what we normally use. And then we get like different peaks from the venom, and we collect each of these peaks. And one peak can contain one, or it can contain, 10 different toxins. And then we test all the peaks in basically the second round. The first round is screening the venoms. And the second round is screening the venom fractions, so the peaks that we're getting. And from one venom, we might get between 50 and 100 fractions. And then if we have identified, okay, fraction number 25 is the one that causes the activity we desire, then we look closely using mass spectrometry to identify what is actually in that fraction 25. And if we can see there's only one component, then we know, okay, it's pure, it's only that component that's causing the activity. And then we can do the next steps in getting the actual sequence. So this is basically like the code of letters, of amino acids. And if you have this sequence, then you can actually produce this toxin in the lab. So you no longer need the spider, which is good because spiders yield very little of venom. And, in each venom, you have hundreds or thousands of components, which means of your component, there's even less amounts in a venom. And to get enough for our testing, we usually need much larger amounts. And we can't get that from the spiders. So that's why we make it in the lab. We can produce it in bacteria or we can produce it by chemical synthesis. And that usually gives us, some milligram amounts of particular, peptide toxins that we are after. But just going back to that screening process. So if it turns out the fraction that we isolated is not yet pure, so there's another 5 or 10 components in there, then it all goes back to this HPLC and we run it again using a different method and try to pull it apart further until we end up with one fraction that we know is pure and there's only one component in there. And then, we follow that up. And as I described, then we identify the sequence. And based on that, basically from that point onwards, we don't need that particular spider anymore for this project because we can just rely on our knowledge of the sequence of this toxin. But this is kind of a bit of a bottleneck in our studies because we need enough venom to get to that stage. So we have a pure fraction that we can send away for sequencing and then get the complete sequence. And if we don't have enough venom to start with, we might only get incomplete sequences. And then, we might not be able to produce the toxin. And if we can't produce it, we can't study it. So there's a lot of sort of challenges along the way. So if all goes well, it's a straightforward process, but on each step there can be, challenges and hurdles that you have to overcome. And that's also why research, takes so long. I mean, some people, that are not involved in research, they always wonder, why this can't be progressed any faster or whatever. But it's also, you have to remember we're studying venoms, that no one has looked at before. Like I actually have 870 different venoms in my collection, and probably 90% of them, I haven't never ever counted it, but I would estimate 90% of them, no one has ever looked at. And so we can't really rely on any protocols, that previous researchers have done because we are the first, like in the case of the spider behind me, the Tasmanian cave spider, no one ever studied their venom before we did. And that happens quite frequently. So we are often the first to look at a certain venom because also, like many people don't have access to the venoms that I have in my collection, so they can't study them. And it's also not that many researchers around the world that do spider venom research. So it's only a handful of people that are focused on that. And usually they do study venoms from their own country. Like if it's the Chinese, they study like local Chinese species, or the Brazilians study species from the Amazon or whatever. But I look more at at these spiders from all around the world that are then kept as pets in Europe. So this is kind of a bit of an advantage that we have. So yeah, anyway, so just saying there's a lot of, like challenges, and that makes our life kind of interesting. You don't have sort of the same challenges all the time. So every time, you look at a new venom, there might be different challenges, but like overcoming these is sort of what makes science interesting and exciting, I guess. It's not like working on a production line where you do the same tasks over and over again.
Yeah. You mentioned the huntsman spider, which here people know as the daddy longlegs. That's the same thing, right?
Not really.
Not? What's the
Um,
daddy? The daddy long legs is not the huntsman,
no, no, the huntsman is like, so normally like the daddy longlegs are the ones that have sort of a bit of a round body
which
and
is
very long,
not even a
thin
spider,
legs.
right?
It depends. So there is a real spider, like a Falcidae is the family. So they have a web, and when when you touch their web, they start making this shaking them as daddy long legs. But then there's the opiliones, so the harvest men, and these are
Harvestmen,
not real spiders. So that's like,
right? Okay.
yeah. So the harvest men are like scorpions, they are arachnids, but this is a different class of arachnids. So the real spiders that have silk. The harvest men, they don't have silk and they don't have venom either. And some refer to them as daddy long legs. And there's even a fly species, so which is an insect, totally different, which also has sort of a small body and very long legs. And some people even refer to them as daddy long legs. So the term daddy long leg is not really, like a unique term for a certain animal. It depends on who uses it. And yeah, but so huntsman spiders are usually huge spiders. And um, they don't have webs, so they run they run on the ground or on a tree or whatever, and then they hunt the insects, while running and while catching them, rather than waiting for them to come into a web and um, be captured by the web. And
Okay.
some of them, they can be like bigger than your hand, like huntsman spiders are. So I don't know if there's another term. I just know huntsman.
Okay, but I did confuse that. I think I was talking about the harvestman. It
Yeah.
has one body part and then a bunch of feet.
Mm.
And it's interesting you said it has no venom, and the folklore around here is that it's actually the most venomous spider on earth, and it just can't bite you because its mouth is so tiny. Have you heard that before?
Yeah, yeah, yeah. We also have that urban myth
Okay,
here
I'm
in
glad to settle
Australia.
it. Here I have an expert to let us all know that this is
Yeah.
out of context. Okay,
Yeah, I
so
mean,
what?
even the spider, where I said that has the web and that does this shaking movements, which is also termed daddy long leg. I mean, that is a real spider, and that does have venom, and the venom has been studied, but they didn't find any components, that would be harmful to humans, and also, there's no clinical record of these daddy long legs being harmful to humans. I mean, that's normally, the first indication. If you have people coming to hospital, bringing the spider and telling you, okay, I've been bitten by this, and then they show some severe symptoms. So then okay, maybe that spider causes some nasty effects in humans, but there's no record whatsoever on these daddy long legs or on these, falsid spiders doing that. But I have sort of a bit of a suspicion where that myth comes from, at least, in my backyard. Because I had like a compost, or I still have a compost bin. And initially, when I opened the lid, there were a lot of redback spiders, so black widow spiders, sitting in the lid, and they eating all the insects in the compost bin. And then after a few weeks and months, they have been replaced by these falsed spiders, by these daddy long legs. So eventually there were no more redback spiders and only the daddy long legs. And if that happened, more often around the area, then people might have thought, okay, I mean, black widows, they're really dangerous to humans, and if there's a spider that eats them, that must be even more, venomous, dangerous, because otherwise it can't eat them. But it's not so much the venom that they're like, that is kind of superior. I'd say it's more the technique of catching the prey, because as I said, these daddy long leg spiders, they have extremely long legs, and they use these legs to pull out the silk from their silk glands, and then they throw the silk basically over the prey. And because these legs are so long, when they catch another spider, like a redback, sorry, black widow, they are so far away from that spider that they can't even be, bitten by that spider because their legs are so long. So it's more like the catching strategy that's advantageous in these steady long legs. And maybe because they can overcome, even larger spiders and venomous spiders, maybe people thought, oh, then they must be even more venomous. But that's not the case. So for humans, they're totally harmless.
That is super interesting. So now on to the honeybee. What is your knowledge of honeybees overall, or is it specifically that you understand the Varroa destructor mite, which I also understand is an arachnid, uh, is potentially destroyed by the venom? What's your interest in bees?
So I'm yeah, I did keep some bees like in my backyard for a few years. But eventually, I think it was last year, they both hives succumbed to the small hive beetle, unfortunately. So now I'm no longer a backyard beekeeper. um, and since we got the Varroa incursion into Australia, I haven't gotten any new bees for the moment. But eventually, I do want to get back into keeping bees again. But one of the reasons, for me keeping the bees, except for having the honey, obviously, is also that, like, besides the varroa mites, my group is also interested in finding treatments against the small hive beetle. Um, so I didn't really, like, treat the bees against the small hive beetle, and because I wanted a certain population in my hives, so I can always harvest them for our research that we're doing. And we already have found a toxin as well, which is sort of which works against the small hive beetle larvae by injection. Um, we haven't tested it for oral activity yet, but so this is something that we are interested in, and yeah, so that was the reason I maybe didn't reduce sufficiently reduce the numbers of hive beetles in my beehives, and then eventually the numbers became too much, and then they died, and I didn't check them for a while, so I was busy doing other stuff, and then they were gone, unfortunately. But yeah, I mean, I do like keeping the bees. It's just sometimes, I didn't have enough time to look after them. And then, yeah, eventually, now I'm without any hives for the moment. But yeah, so the interest in the Varroa might actually started much earlier than me keeping the bees. So that was basically, among all the other pest species that we're looking at, I realized quite early, that Varroa mite is a significant problem, and I started approaching experts in that field, like in Varroa mites and bees, already in 2016. And just as a reminder, the Varroa mite came to Australia in 2022. So that was 6 years before we actually got Varroa. I started, my project, and I got together with an expert in Switzerland, in Bern. Vincent Dietemann is his name. So I don't know if you know the Colossus bee book. So he's one of the lead authors on the Colossus bee book. So this is about like ways of keeping bees, and how to manage pests and parasites and stuff like that. I mean, he's sort of one of the world leaders in research on honeybees. And he was also knowledgeable about Varroa mites. And because at the time when I started my project, we did not have Varroa mite in Australia, so I couldn't test them in my lab. So I needed someone doing the testing for us, and that was Vincent. And so yeah, he basically harvested the mites from the hives they had in their research institute, and then he did the testing for me, and I just sent him the venoms. And after he identified which venoms were active, I sent him the venom fractions to find out which peaks caused the activity and so on. And the main challenge in that collaboration was that in Switzerland, in Europe, these varroa mites are usually, I mean, all the life cycle of bees is kind of seasonal, right? in winter there's not much activity. They just kind of rest, waiting for the temperatures to get higher, and then the bees, start increasing in their colony size again and fly out. And it's also when they start sort of increasing their numbers. This is when the Varroa mite numbers increase. So which means you don't have sufficient Varroa mites for testing all year round. So it's mainly during early summer, or then maybe to late spring, and then sorry, late autumn. And so it's only a few months a year where you have sufficient Varroa numbers available. And that made the project very challenging because, at the start of the Varroa season, I needed to have enough samples sent to Vincent so he can test throughout the entire season. But if we want to follow up any hits, we often had to wait till the next season. We might have to wait for another 6 months until the Varroa mites became available. And this is why the whole project took so long, from 2006 until we eventually published that paper in like this year, 2026, so 10 years later. Because, like these challenges, I mean, if you don't have Varroa for testing, you can't simply do the testing. And yeah, it might be easier now with the Varroa being in Australia, because I guess especially in the more tropical areas or subtropical areas where the bees are more active all year round, you also have more easy access to Varroa mites. But yeah, so far, all the tests that we have done and that we have published, they have been carried out by our collaborator in Switzerland.
So how do the Varroa mites succumb to the venom?
So the way we have tested them is basically we flip them on their back, like on a bit of a glue strip, and then applied the venom, like little drops of the venom, so like a solution that contains the venom, onto their belly, onto their underside. And then basically waited for a certain number of hours, so like up to 2 or 3 days, and then checked, how are the mites behaving? Are they still alive or are they dead? And we've done that, for mites that have been treated with the venom, but also for mites that have been just treated with like water or control solution. And then you compare, which of them stay alive longer. And so what we found from our experiments is that like even the mites that are treated and tested this way, with the control solution, after 48 hours or so, all the mites will be dead eventually. Because if you put the mites upside down on a rubber strip and you don't give them water or any food, eventually after 2 days they will die. But what we found is that when we applied our venoms and later on the toxins we isolated, they die more quickly. And so we could do statistical analysis and say, okay, this is not just a random effect. So this is a real statistical significance. So they are dying more quickly, which means our toxins are active. And sort of this is kind of the main or one of the most important findings of our study, that we can kill the mites more quickly than they would die of kind of natural causes, or when you just let them starve. And we also used, as a control solution, we used oxalic acid as well, and that obviously also showed a much quicker response than the control solution. So the oxalic acid, after a few hours, like after like 12 to 16 hours, all the mites were dead. And for our best toxin, it took a bit longer. So it took like 1 or 1.5 days till they were all dead. And for the control, it took like 2 days until they eventually die. So we can see there's a clear difference in our, like, what our toxins do. And also, the oxalic acid was quite potent. But this can also be like, it was in our assays, it looks like, it's acting more quickly than the toxins that we have found. But that might just be a dosage issue. So if we use a higher dose of our toxins, they might also have a quicker effect. So we do see that often. If you increase the dose, you will see the effect, speeds up. But we only had a limited amount of the peptides for testing. So we tested at a certain dose, and at that dose, they were a little bit slower than the oxalic acid, but they still showed the activity. And then after proving, our toxins that we isolated from the two spiders, it was not just the Tasmanian cave spider, it was also the giant Japanese funnel web spider, which are not related to the funnel web spiders we have in Australia. So they're not dangerous to humans. And so the two toxins we isolated from these spiders, so they show the activity in the Varroa mites. But then we needed to ensure, they don't kill bees as well as they kill Varroa mites. So we have done kind of the same experiment where we used honeybees, and then we applied the solution of these peptides topically to the honeybees to see, how they behave. And then we observed the honeybees for much longer time, because in the assay, they just survive for longer. So we observed them for up to 20 days. So eventually, even in the way we house them and we keep them, in the lab, after 20 days, all the bees will be dead. But we didn't see any increase in the time any decrease in the time it takes the bees to die by any of the treatments. So the the control solution or the toxins that we applied, they didn't change the time of killing the bees. So which means the treatments kill the mites, but they don't affect the bees. And this is kind of what we want. And but this is only sort of one safety measure, obviously we need to ensure, the bees are safe. But obviously, the beekeepers also shouldn't be harmed by any treatment that we apply. And but obviously we can't do experiments in humans. But we have used some proteins which are called ion channels. So these are important proteins that are involved in the transduction of electrical signals in the human body. If you don't have them, you can't send, for example, signals from your brain to your muscles or to other parts of your body, and your body does not work. And if these toxins interfere with this transmission of electrical signal, they could potentially be harmful to humans. But so we didn't find, for our toxins that we tested and the ion channels that we tested, we didn't find any significant effects on these human targets. So based on our testing so far, there's no concern about human off-target activity. But obviously, if this became, like a commercial product, eventually, we might have to do more extended off-target testing. So maybe, injecting it in, rats or mice, maybe, and trying some, like in vivo experiments, not just on ion channels, also trying, on real vertebrates like mammals, and see whether it has any off-target effects. But so far, from what we can tell, these two toxins that we identified, they should be safe for humans, and they should also be safe for the bees. But hopefully, they will be good to kill the Varroa mites. And that's sort of our idea. And now the main challenge is sort of how do you apply it in the hive, right? Because we had the individual mites and the individual bees for testing, and then we could just use a small syringe and apply certain quantities, defined quantities of our material onto them for testing. But apparently you can't do that, in a hive, where you have tens of thousands of bees and some of them, carrying the mites. So, and we figured, the easiest solution would probably be like a spray, where you can spray the hive, and then maybe before the night, you spray the hive, then close it off overnight so it can sit in there, for a few hours, and then when you open it during daytime, hopefully all the mites will have fallen off and the bees are still happy. So that would be the ideal solution. But we haven't tested that yet. So I can't say whether it works or not. And this is sort of what we're working on at the moment. So we got funding from the Community Bee Innovation Fund here in Australia. So we recently got like $50,000 to do some follow-up studies on our two venom peptides. And this is what we're planning on doing. So first of all, we want to still do some lab testing where we have the mites that actually sit on the bees rather than being separated from them. And then we spray them both at the same time, and then we see how that works. And if we can show, okay, this works and the mites drop off dead while the bees are still alive, then we can go the next step and say, okay, now we produce even more of our peptides, and now we apply it in a few hives and see how that goes, and then compare, the numbers of mites in the treated versus the non-treated hive. But this is still sort of what we can do maybe in half a year or a year. So first of all, we still have some more initial steps in the lab, and we also need to produce larger amounts of these peptide toxins. Obviously, for our research so far, if we had like 3, 4, 5 milligram, that might be enough for the initial lab testing. But now, if you want to apply it more in a hive, you need much larger amounts, and we first need to produce them in the lab, which also takes a few weeks. So it's nothing you just do in an hour or so. So because you need to produce it and you need to purify it and ensure you don't have any contaminations that would affect your activity, and so that's all a longer process. And this is also another reason why we need constant investment in research, because without doing these studies, we never know, and we can't whether it will work eventually or not. And so we can't develop any commercial products. We don't have continuous funding, and that's a particular challenge, not just for this project, in general. And I see this all around the world, many countries, they reduce the money they put in research funding. And if you don't fund research properly, then, you can't get any benefits that come out of research. And and this not just affects the beekeepers, this affects all areas of life, basically. And I just see it sort of as a challenge, especially for my lab, for example. At the moment, I do have 2 postdocs that work for me and on this project and on other projects. But I only have funding for them basically for the next year, and then we don't know what happens thereafter. So for if you like a postdoc, my position is permanent because I also do teaching and research, so I'm employed by the school. But my postdocs, they are employed, by the university, but in my lab, and I have to pay for them through, research grants that we are, for example, getting, through this Community Bee Innovation Fund or through the Australian Research Council. But I constantly have to find this money, and if I run out of money, then and if my postdocs are not getting a salary anymore, they might say, okay, I have to go somewhere else because they need to pay rent and they have living expenses, so they can't stay in my lab and work for free. And then, I'm losing their precious, skills and knowledge that they have in, target treating or testing certain pest insects. And then if I eventually get more funding, 2 or 3 years later, then I have to hire new staff and teach them again because they don't know what my current staff knows. And so this continuity is very challenging in research, because you always work from one project to another, and there's never the guarantee, that after the end of the project, you get the next one, and the next one, and the next one. You might, your funding income might run dry for a number of years, and then you have to reduce your staff members in the lab. They go somewhere else where there is still money. And then when you eventually get money, you have to rehire staff, and that's obviously a big sort of brain drain, for each lab. And also on a more national scale, obviously, if certain countries decide, or will no longer invest in research, then the researchers just decide, okay, I go someplace else, where research is still more valued and paid. And that also happens, around the world. And so this is challenges, that are just outside, the Venom research that I'm doing that more generally affect research all around the world. And I'm lucky that I got now like a permanent position, but I've been postdoc for 15 years, and I have been in that situation where I know, okay, like my contract is up for renewal, in a year, and I don't know if my supervisor still has enough money to pay me. And this is always a very challenging time for people in that situation. Mm-hmm. And I think more money is definitely needed in research. But the problem is, for research, the benefits usually come after, let's say, 5 or 10 or 15 years. You find something now, but until it translates into a product, into an actual benefit for humans, that takes quite a long time. But then, when you look at politicians, I mean, their cycles are usually like 3 to 5 years. So they want benefits immediately, that could benefit them in their political career. For a politician, there's no benefit in having a huge, success from a research, in 15 years, because they're no longer in their position during that time. And I think that's a big problem. So the people that decide like on how much funding is used for research, they're not interested in the long-term gains. They're more interested in the short-term gains. And you often don't get that from research. And this is a big problem around the world.
And so what capability is new? In other words, your ability to render this Venom, replicate it, is that something that you could have done even 10 or 15 years ago? Or do you have new abilities available to you that previously you couldn't have done it?
I mean, obviously, we're getting I'm increasing my Venom collection every sort of 2 or 3 years, when I go on another collection trip. So, I mean, like 10 years ago, obviously, like the Venoms we had available were a lot less than what we have now. So maybe, there might be other toxins in some of the newer Venoms that I have that are even more potent and suitable against the Varroa mites than the ones that we have screened. But to find that out, I would need sort of another research project and start again, initially just using the new Venoms and screening them and seeing if there's any Venoms that work better than the ones that we have already tested. And this is sort of new capabilities, obviously, that was every year, every few years, we're increasing our Venom biobank. But it's also in terms of, like the technical development. I mean, like some research equipment that we use, like, mass spectrometry, they get more sophisticated, every year, and with like quantities that we can use nowadays to detect some signals, like 10 years ago we wouldn't have been able to detect them. So we get better technical capabilities, obviously, and also, I mean, more recently, like the use of AI is also sort of a tool that helps us in our research, because we can sort of get now like structures of these peptide toxins, where before we had to do sort of more complicated experiments to determine a structure. And nowadays there are programs that you can use, and they can predict your structure just based on the sequence of your peptide, and based on the known sequences and structures of other peptides. They have like these huge models that know sort of all the published sequences of peptides and proteins and how their structures look like. And based on that, they can predict how your peptide will look like. And before, that was a quite a difficult sort of experiment, and where you needed machinery, that costs like a NMR spectrometry, for example, where the equipment costs, between like $500,000 and $1 million. So you needed access to this expensive equipment to do this experiment. And nowadays, you can predict your structure on a computer at home if you have the right program. And so there are certainly, ways in which research, improves every year, which is good. It's like it's never a process. There's never a standstill. So you always have to be, up to date on what's happening in your field and new developments and how you can potentially, how a new development maybe in another field like AI can benefit your own research. So it's always like a constant development, but there's not sort of one key thing, where I said, oh, this made sort of a major breakthrough or whatever. So it's sort of little bits here and there that always help you, getting better. But then again, if you wanna publish your results, obviously, like the editors from the journal, they also expect more from the studies nowadays than 50 years ago. So a paper that has been published 50 years ago with the methodology that was used then would never be accepted nowadays, because now they know there's more sophisticated equipment out there and where you can get a lot more information. And so not all old papers are no longer valid, but many of them, you couldn't publish nowadays because the expectations also increase with all the increase in our knowledge and the technical sophistication.
Mm-hmm. Now, I realize you're going into practical beehive testing. What do you anticipate, if any, the risk to be with exposure to honey and what people might be eating out of the hive?
I don't see any issues for that, because like what we are using are peptide toxins. So peptide is basically like a little protein, and they are built of amino acids. So it's basically like a chain of amino acids. And amino acids are the building blocks of life. So, all our bodies, all the proteins in our bodies are made of amino acids. And these toxins, if we can show them, they're not harmful to humans. So what will happen, even if they would accumulate in the honey, so eventually the enzymes in our body, they would just break them down in the individual amino acids. And then these amino acids could be used to build up other proteins in your body. So like the degradation products of these peptides are kind of harmless, and they can be reutilized in your body to build other stuff. So it's not like, there's any harsh chemicals, that could cause any, severe symptoms in humans. I wouldn't be concerned about that. And then we have done other experiments with some of these peptide toxins, where we can, for example, show, like for the ones that we're planning on spraying, on crops. So if you expose them to sunlight, so especially the UV radiation, after a few weeks, they will naturally break down into the amino acids. So they're not as super long lasting as some of the chemical insecticides that we have been using, in the past, and that have accumulated through the food chain. So there are enzymes in our body, that can digest peptides and proteins. So I wouldn't be scared of that. And so if we can confirm, there's no harm directly of the component that, like when the humans eat it or when it gets into a human body, then I wouldn't be concerned. And this is something, we'd have to test anyway, like to ensure, or at least, in some mammals, there's no off-target activity. And then eventually these compounds would be broken down. And like if you eat them, like by consuming honey, so most of them, they would just be like digested in your stomach. So they wouldn't go into your blood or to be able to, to affect your nerves. Because this is where most of these spider toxins like the spider toxins that harm humans, they're neurotoxins. So they affect the nervous system. But, they can only affect the nervous system if they get to the nervous system. But if they're digested in the stomach, they never get to the place where they could potentially be active. So I'm saying, even if these toxins would be toxic to humans, you could probably still eat them and be safe, because you can't take them up through your stomach. So that's another advantage. But obviously, we try to use toxins, that are harmless in the first place, so we don't have to worry about. But yeah, there are additional sort of safety measures for these peptides, and I wouldn't be concerned at all, of any off-target effects. We just need to ensure, they're stable long enough to be active. So if they're, if they stay active for, a day or two, that should be enough to, spray your hive and then kill the mites, and then they can be broken down by natural enzymes or by sunlight. When the imagine, you spray a hive, and then obviously the toxin covers all the bees and the mites. And when the bees then fly out to the flowers, so eventually, they will be exposed to sunlight. And this way, this is where the peptides will break down eventually. And yeah, so I think the safety is really good. So there shouldn't be any concerns. I mean, what we have to worry more about is, resistance development. So we do see that for some of the chemical treatments that are used nowadays. So like in Australia, they have now found mites that are resistant to 2 different classes of chemical insecticides. So they call them the super mites, which is obviously difficult for beekeepers, because they no longer have like a good arsenal to treat their hives. And these spider toxins, they would not be safe, from resistance development, because that's just like a natural process. Any treatment that you give, to an organism over and over again, eventually, they will build up some way of resistance. That's just natural evolution. Eventually, one of the organisms will have a mutation, and that no longer allows your compound to be active. And then, this organism then persists and reproduces, and then eventually you end up with a population of organisms that are no longer susceptible to your treatments. And the same can happen for the toxins from the spiders. And this is already sort of happening in nature anyway. So for all the toxins that the spiders make to overcome their prey, there's obviously a constant like race in evolution, between the prey and the predator. So the spider makes new toxins, and then the prey comes up with new ways of, rendering these toxins ineffective. But then the spider comes up with other toxins that are still effective, and that race goes on and on and on. So that's just nature, that's how evolution works. And so we can't really prevent that. If we, let's say, the spider toxin would become a commercial product and it's very effective, but if we only apply that spider toxin over and over and over again, eventually, we get resistance. That's just normal. So what we have to do is sort of maybe apply it, like spider toxin, and then apply another component and maybe another one, and then go back to the spider toxin. So don't do the same thing over and over again. Mix it with some other treatments. Or what we could also do is co-applying, at the same time, you give the spider toxin and another component that acts via a totally different mechanism. If you have like 2 components acting on different mechanisms, it's very difficult for an organism to acquire resistance, because they might get a mutation that helps them against one treatment, but then the other treatment will kill them. And the chances that they acquire mutations at the same time that helps them against 2 different treatments is very low. So by using combination treatments, that would be much safer. But there's more an issue for these combination treatments when it comes to the registration process, because to register a combination treatment out of 2 or 3 compounds, it's a lot more difficult process in terms of the official registration, because the testing you have to do is a lot more extensive and more costly, because you have to show all your individual components are safe. But then when you apply them together, they're still safe. So you have to do a lot more testing around that. And that obviously is more expensive. And that's why why mostly, in agriculture, when you have treatments, they just have a single treatment, because it's cheaper to develop it than a combined treatment. The requirements, for the registration are a lot less. And and this is sort of a problem I see, and that's nothing that I can sort of solve because I don't make the regulations around the registration for these compounds. And yeah, but my recommendation would be, like if possible, try to use a combination of different compounds or that work well together, or try to mix, like vary which treatment you're using. Don't use the same over and over again. So use one and then the other and then the next one and then go back to the first one. And yeah, that might be a better strategy.
Has the open brood been exposed to this treatment?
Yeah, we haven't tested it on brood yet, and I'm not sure, because like especially when the brood is kept, it was like the wax capping. It would obviously be challenging, like getting our treatment to them. So we need to think of ways, of how the encapsulated brood can be treated. Because if we spray a hive, and if your spray just sticks on the wax capping, then the Varroa mites inside those brood chambers, they would not be affected because the treatment can't get to them. And so yeah, I don't know if there will be ways that we can sort of overcome this. Or, I mean, alternatively, you'd have to give it maybe, like multiple times, do one treatment for the bees that are sort of, for the adult bees that are out now. And then a few weeks later, once all your brood hatched, do another cycle of treatment and then make sure you kill the Varroa mites that have come out more recently. And this way, you maybe reduce the numbers. I guess there will be no treatment, that really kills and eradicates 100% of the mites. So it's always, like managing the mites. It's always reducing the numbers to a level, that the bees can live with. But you never get complete clearance. And even if you did get complete clearance, then one of your clean bees flies out to a flower and meets a bee from another hive that's infected, and then they transfer the mites, and then they bring back the mites to the hive. So yeah, once you have the mites in the country, you can never expect any treatment, that completely eradicates them. It will always be about managing and what is the best way of, keeping the mite numbers low enough for the bees to live a good life, and not letting them, getting out of control and then the hive to collapse. And sort of that's the best, I guess, we can hope for. And to have ways of, constantly managing the hives, we need more and more and more new treatments, because eventually, because there's only a limited number of treatments and people use them over and over again. That means, eventually, there will be resistance against these treatments, and then we need new treatments that are where the bees or where the mites no longer have or not yet have resistance. But if we only have, always a low number of treatments, eventually, they might be susceptible to resistance. So the more treatments we have and the more we can mix and match, the safer we are. So yeah, that's sort of my recommendation. But as I said, this would mean a lot more money into research, and it will take time. It might take 10 years until we get more treatment. So we have to invest in research now so that we're safe in the future. And yeah, we can't just sit back and relax and say, now we found this spider toxin, that's great, and that's all we need to do. So now is the time to look for new treatments, basically, because what works now might not work in 5 or 10 years.
Mm-hmm. Now, I realize this is forecasting also, but the Tropolelaps mite, which is also showing up as a devastating parasite for the honeybee, would you think a similar approach would work for the troping mite?
It's funny that you mentioned that, because we discussed that recently with Vincent Dietemans, my collaborator from Switzerland, who worked with me on the Varroa mites. and he mentioned, so to me, I wasn't aware of that parasite actually, but he mentioned, that's sort of the next upcoming issue, for the beekeepers, and because it's also approaching, I think, like Europe at the moment, and like it's already in Southeast Asia, and so we've been in discussion, with some collaborators here in Australia, and also with my Swiss collaborator, and we found someone in Vietnam who could do the testing for us. So we're planning on submitting a research grant on that. That would involve both testing more venoms on Varroa mites, like newer venoms that we haven't tested before, but also testing some of the active components against the Tropilelaps mites, and see, maybe you might find a treatment that works against both mites, which would be great, then. So yeah, we're planning to integrate those mites as well into our new testing, provided we do get the research funding. But at the moment, like depending on the research grant that you submit, like the funding rates range, between maybe 10 and 20%, which means, at least 80% of applications don't get any funding at all. So it's by no means guaranteed, when we submit our grant proposal that we will get the funding. So we might have to try, a few years in a row until we are eventually successful. And that's the main challenge that we are facing. I mean, we could do a lot of interesting research, but to do that, we obviously need the money. And most of the money, for research, when you see these grants, they might be worth, $500,000 or $1 million or whatever. And people might wonder, where does this money go to? Will it make the researchers rich or whatever? But so most of the time, like 85% of that money will go into salaries. It's just paying the staff members that work on these projects. And then you have a bit of money, for your consumables, for your chemicals you use in the lab, and the reaction tubes and whatever you have to buy for your experiments, for maintaining your insect cultures, the beehives. You might, like in the lab of our collaborator in Switzerland, they obviously have a professional beekeeper who looks after their hive, so the beekeeper needs to be paid. And so most of the money in research actually goes into salary of the researchers that are doing the work. And then, there might also be a little bit of money, so you can publish your results in certain, journals, like in peer-reviewed journals. And there might be a bit of money for travel involved if I want to visit my Swiss collaborator to do some experiments in his lab, or to do some, discussion around writing papers and stuff. Then we need some travel money, or to go to conferences. But yeah, the vast majority of funding is to pay staff, and yeah, unfortunately, there's not enough out there. There's a lot of good researchers, and they all have good projects, but they can't be all funded at the same time. So there's always someone who will miss out, and that's not because of the quality of the research project, but because there is not enough money in research. And I guess that's not just here in Australia. So I can see that in other countries as well, where people are struggling.
Well, I think we've covered the ground pretty good, and I think we're going to be looking forward to seeing what transpires over the next year here with your advancement into practical testing and with real beehives. Do you have a beekeeper that's in Australia that's going to collaborate and give you access to their apiary?
Yeah, I've been in contact with some beekeepers, that where I could potentially, like get bees and mites from. And I'm also, like the Department of Primary Industries, they look after managing, like the Varroa mites, and they know sort of beekeepers that reported, infestations of Varroa in their hives. So because, we obviously want access to hives that are infested. So we get both, like the bees and the mites at the same time for testing our components. And so we're working together with the department to find the most suitable beekeepers, in our area, obviously around the Sunshine Coast, so we don't have to travel too far. So we can easily go to maybe the closest beekeeper in that area and then grab some bees for testing. And then they also have the mites attached, and then maybe grab some, brood frames as well. So we can harvest more Varroa for experiments. And that's sort of the plan for the next few months, after we, produced enough of the peptide toxins for all our testing efforts. So this is happening right now, so that the peptides are produced in the lab and purified. And once we have that, we'll start the testing and get in contact with some beekeepers. So yeah, maybe we can do like an update on this in a year's time, and hopefully by then we'll have some more interesting data to report, and hopefully some positive data. But in research, you never know. Normally you make also for your research grants, you make like a big plan, what you want to do in the next 3-4 years for your research project. And then like you do your first few experiments, and then you have to change everything because it didn't go. You can't always know what the outcome will be. You can assume, like this might be the outcome, and based on this, we do the next step. But often, like your experiments don't go as planned, and then you have to change all your plans. And so we never know, like we can only hope the project will go according to plan, but whether it works or not, only the experiments will tell us.
Mm-hmm. Well, I want to thank you very much for your time and for sharing so much of what you did. And I'm sure the wheels are turning for a lot of people that are listening. And if you want to look down in the video description, there will be a link to the 2026 published study. And any updates that I receive, of course, will be added as a resource for those of you who want to do further research and learn more about what's going on with this grant. So thank you so much for your time. I really do appreciate it.
Yeah, you're welcome. Okay, have a good day then. Bye.
Okay. Yeah. And that wraps up another episode of Interviews with Experts. Please don't forget to visit the description for additional information and links that you may find helpful. I'm Frederick Dunn, and this has been The Way to Be Interviews with Experts.